Idiopathic pulmonary fibrosis, or IPF, is a chronic and progressive interstitial lung disease. It typically presents in individuals older than 50 years, and more often in males. IPF occurs worldwide. It is a rare disease, but its incidence appears to be increasing. The exact cause of IPF is unknown. However, it has been linked to environmental and genetic risk factors. The most common symptoms of IPF are a persistent cough and shortness of breath, and these progressively worsen over time. It is ultimately debilitating and has a poor prognosis. During respiration, oxygen is taken into the lungs and carbon dioxide is expelled. An important step in this is the diffusion of oxygen across the thin interstitial space between the alveoli and surrounding capillaries. IPF is characterized by a progressive scarring of the interstitial tissue of the lungs, which is thought to be the result of abnormal repair processes in the alveoli. A feature of the abnormal repair is exaggerated expansion of myofibroblasts, which secrete extracellular matrix. Additionally, an increase in profibrotic macrophages are observed in the alveolar space. Studies suggest galectin-3 plays a key role in this process. Galectin-3 is a carbohydrate-binding lectin that has been shown to be highly expressed in fibrotic tissue. Galectin-3 can localize on a wide range of cell types, which includes macrophages, fibroblasts, and epithelial cells. Here, on the cell surface, it can potentially form lattices and aggregate receptors. This retains the receptors on the surface, which amplifies profibrotic signaling. Galectin-3 can bind to a range of receptors. These include the transforming growth factor beta, or TGF-beta receptor, and the platelet-derived growth factor, or PDGF receptor. The signaling can lead to progressive interstitial thickening, which impairs gas exchange and respiration. GB0139, also known as TD139, has been developed as a dry powder for inhalation. This allows targeted delivery and has been shown to provide prolonged lung retention. GB0139 is a small molecule galectin-3 inhibitor that is hypothesized to interfere with signaling on the TGF-beta receptor and may have effects on multiple other receptors. This results in disruption of the galectin-3 lattice and receptor internalization. This potentially limits profibrotic signaling, which may prove to be clinically efficacious in reducing IPF disease progression.